professor at MD University, Rohtak, and currently, after working at JCDM College of Pharmacy, Sirsa as a professor, he is working as a professor in AMITY University. He has supervised 4 PhD and 40 MPharm students. And currently he is supervising 5 PhD students. His key research areas are medicinal chemistry, molecular modeling, chemoinformatics, nanoparticulate drug discovery, analytical method development, and he has a research expertise in bioinformatics and chemoinformatics tools for structure and ligand-based drug design like docking, pharmacophore modeling, QSAR, molecular dynamics, and homology modeling. He has completed drug discovery projects on cancer, diabetes and metabolic disorders, as well as Alzheimer's and autoimmune diseases. He has to his credit about 95 national and international publications. His citations are 1800 plus. Google Scholar H index is 20, I10 index is 36, and Scopus H index is 18. Cumulative impact factor to his credit is greater than 150. He has written 9 book chapters published by international publishers, has many research grants, and is awarded with prestigious awards like Jubilant Biosys Limited Award, Appreciation Award. He is a recipient of the gold medal in the MPharm and has really done very well in his academic career. So it is our pleasure to have Dr. Vinay Lathar as a first speaker of the third day of 32nd Quality Improvement Program. Dr. Vinay Lathar, please.
Yeah, so thank you so much, ma'am. Dr. Amrita Parle, I'm extremely thankful to you for the nice introduction. So at the outset, I would like to thank the DIPSAR, the entire DIPSAR family, especially Ma'am Parle, Dr. Sharad Wakode, and Professor Pathak for giving me the opportunity to share my research experience, which over the years I have gained. And most of that experience probably which I'll be sharing is in the field of the virtual screening, basically the computer-aided drug design tools, which can be used to fasten the process of drug discovery. So I hope I am very much audible.
Yeah, you're audible, sir.
So okay, thank you. So I'm just sharing my screen and.
Sir, before you go ahead, if you can increase your volume a little, that would be great.
So I hope my screen is clearly visible.
Yeah. Screen we are able to
so
yeah, screen is visible, sir.
Sure, ma'am. So we will be talking about the different virtual screening or the computational tools which are being used in the drug discovery. So probably I'll try to finish up my presentation in like around 45 to 50 minutes, and then I'm sure that there will be a lot of queries from the participants. Since probably since everybody is probably nowadays working in the field of these computational tools. So whatever they kind of drug discovery projects they are working. So that's why it's better to have a one-to-one discussion for 10 to 15 minutes and take the different queries from all the participants. So if we look at the drug discovery, so I'm not going to talk about the drug discovery process, which takes like 15 years of time, and the cost of the drug discovery is more than billions. of dollars. And so let's not I'm not talking about that part today. So let's focus on the another thing, which is like how the drug discovery has been evolved from the phenotypic drug discovery approach to the target drug discovery approach. So earlier, if we go back maybe around 50 years back or something like that, so this was the traditional approach which was being used, where the phenotypic drug discovery approach and the targets, they were unknown. So the receptors of the proteins, the drugs which are working on, they were not known initially, and we were like discovering the molecules based on the studies on the native human cells or the animal cells, on which we do induce the diseases, and then do the experiments and test like which are the which compounds will be effective. And later on, depending on that, then we used to go for the need to for the deconvolution to identify the target in the last step. So, but now over the years, the drug discovery has changed in a tremendous way, and it's more like kind of a target-based drug discovery approach, where we begin with the targets, the individual receptors on which the drugs are acting. So for each disease, we identify the specific targets, we validate them, and then we develop the screening assays. And mostly like we use the recombinant proteins or cells over expressing the target of the interest, and then we use the high assay throughput screening, so high throughput screening methods, which basically decreases the time of drug discovery. So the screens which are used to measure the compound's efficacy on the target of interest, they are also known. So need to confirm the compound's effect in the biological effect ultimately. So look at if yeah. So over the years, if we look at the vast expansion in the field of chemicals as well as the biological targets, so there is a huge explosion of data. So as of now, we are moving in every field towards the artificial intelligence, and artificial intelligence, we say like it's basically based on the accumulation of the data and processing that information, and ultimately getting the kind of the deemed whatever the intentional results. So that is, we are moving towards artificial intelligence, and how we are moving that artificial intelligence into the drug discovery, that also a little bit I will focus in the end. So if we look at the data explosion, so we have approximately in the chemical abstract services presently, if you look, so this is the data of 2015, where the data, we have approximately 162 million entries. Okay, so this is the like approximately 162 million molecules they were reported in 2015, and this still the data is expanding, and I hope by 2020 it will be like still more than it might have crossed the 200 million entries. And if we look at the Cambridge crystallographic database, so it's like more than 1 lakh, more than 1 million of entries are there. So we have determined the crystal structures of approximately 1 million of molecules, and so that gives a kind of a huge chemical space which is available as a starting point for the drug discovery projects. And similarly, if we look at the kind of the genetic information which has been processed after 2002, when the Human Genome Project was resolved, and we are very much aware, like nowadays, like more than till the 2018, 200 million sequences, 200 million nucleotide base sequences, they have been identified. So probably many of you might be aware that human genome, which is like it's a genetic makeup of a human being. It is containing approximately 30,000 genes, and those 30,000 genes, they are ultimately made up of 3 billion base pairs of nucleotide sequences, and each of this gene, 30,000 gene, is ultimately responsible for approximately 3 to 4 protein, is making the 3 to 4 protein sequences. So that's like a vast expanse of data is available in the terms of genomics as well as the proteomics. And now how we are going to utilize this kind of information for the drug discovery. If since this COVID-19 pandemic, the things have changed, and most of us, most of the drug discovery, which was focusing on kind of cancer and other CNS disorders, autoimmune disorders, but those things are like in the kind of a break is there in that kind of research, and now everybody is focusing on the treatment of the COVID-19. So, but overall, if we look, so before that, if we look at the point, so there was like kind of a very much interest in the precision medicine also, where we are developing where we are going to develop the specific medicines for depending on the genetic sequence of the individual human beings. So we are kind of going into that direction also, and probably in the next couple of next 2 decades, we will have a kind of a system where we will have a system in which you will go to your doctor and he will be having your complete genetic sequence, and based on that probably like the proteomics information also. So what kind of medicines will suit you, he can prescribe you. So this kind of an information is like really helping us and going to change the drug discovery in future. And also, if we look at the number of the crystallographic information of the proteins which have been entered into the protein data bank, it's like expanding phenomenally. So at present, if you look like, so it's approximately 160,000 entries are there, which are available as the drug discovery targets, and we can use that information for the drug discovery. So as I told you, so we have a big challenge in the drug discovery, that we have a very huge chemical space which can be used for the drug discovery, for the discovery of new drug molecules. And similarly, we have a huge biological space or the target space, which is like large number is available. So we have to ultimately result into a right molecule and the right target. So that is ultimately the aim of the drug discovery. So probably like you all are also very much aware that most of the drug discovery projects which are being carried out by the pharmaceutical industries or even in the academic research nowadays, what exactly we are doing, we probably go into taking a library of molecules, and then we go for the screening of those molecules against different types of targets, and then develop the biological assays and use that information ultimately for the in kind of animal models. And then we go for, if all things are going into right directions, then probably we are going for the clinical trials and those kind of studies. So how we can reduce this time and how we can choose the right molecule. So ultimately, to choose our goal is to find a lead compound that can be optimized to be a drug candidate. And the optimization is basically using the chemical synthesis to modify the lead molecule in order to improve its chances of being a successful drug. So the challenge which is lying in front of us is like huge chemical space. So huge chemical space, like there are 1 billion compounds with up to 13 heavy atoms are available. There are more than 30 million known compounds which are available. And the typical pharmaceutical compounds collections contains 1 million compounds. So if we look at the kind of the libraries which are available, so with the different organizations, so like NCI is a database which is containing a million of molecules. Similarly, we have the Affinex database. Even we worked at the Jubilant, so we also used to curate the data, and that data we used to make depending on the kind of like which category the drugs belongs based on the receptor. So we used to create the data based on the public literature. We used to get the information from there, and then we used to make our own library, like the GPCR library we developed there. We made a library of ion channel receptors. We also made the library for the especially for the kinases enzymes, which are the potential targets in the as anti-cancer agents. So those things we were then developed. We developed our own libraries also. So, and the last thing, high throughput screening can be used basically for the large number of compounds to be tested, but the problem lies in there that very small proportion of available compounds. So the number of available compounds with each of those pharmaceutical organization is less. So we need to either buy those chemicals or we have to synthesize those chemicals. So that is another problem, and it will increase the time of the drug discovery. So large scale screening is sometimes it's more expensive if we go for the screening of millions of compounds or billions of molecules, and not all the targets are suitable for high throughput screening. So sometimes we are not able to develop the assays for the targets which we are looking for. So that's why we turn to another area which is like computer aided drug design. So in computer aided drug design, most of you might be aware also, like this is kind of a two types of approaches which are being used. So one of the approach is called the structure based drug design, and the second one is the ligand based drug design. So in case of the structure based drug design, again there are two opportunities when the structure of the target or the structure of the protein is known, and the crystallographic information, or we can say the 3D coordinates of the structure, they are available. So we can use that information for the ligand docking. We can use the molecular docking as the tool, as a virtual screening tool. If that the ligand information is not available, so we can use the de novo design approach, where we use the binding site information for the design of the new molecules. So the another approach, so the unknown, if the protein structure is not available, so in that case we will be going for the ligand based drug design. So in that ligand based drug design, like the chances are whether we are beginning with the one or more ligand is available. So what kind of approach you will be using? So we'll be using the similarity searching kind of an approach. If the several ligands are available, so we will be using the pharmacophore searching or pharmacophore modeling approach. So most of, if there are many ligands are available, so we will be using the quantitative structure activity relationship. So if the ligand structures are also not available, so in that case, what is no ligand is also not, no ligand is available in that case. So the computer aided drug design will be of no use. So in that case, we have to be dependent on some kind of an experimental data, and then we can apply later on the some admit filters. So those can be used basically to decrease the time of the drug discovery. So overall, if we go to the virtual screening, we have a large chemical space where we have billions of molecules are available with us. So what kind of approach we will follow? So in the beginning, we'll be using some kind of general filters. So like we can use a kind of a substructure approach. We can use the drug-like filters. We can use the admit filters. So these are the some of the filters which will eliminate some of the molecules in the beginning, and then a large number of molecules basically it will eliminate which are not fitting the parameters, like they don't have the characteristics like drug like parameters, so they don't have the drug likeness. So in that case we will eliminate these things based on these kind of filters. And then what next approach we can follow, the ligand based approaches, where we'll be using the similarity searching, pharmacophore modeling, quantitative structure activity relationship, and nowadays like another approach which is rapidly emerging up, which is like machine learning. It's not a new approach, but it's being used since last 15 to 20 years. We are using like support vector machines, neural networks, so kind of supervised and unsupervised learning methods to screen, to classify the molecules based on their activity profiling, whether they will be active or inactive, and then if they are active, so we can screen them against the different assays which we have developed for the particular diseases. And the last approach, which is like docking and the de novo design, and also the free energy perturbation calculations, so which ultimately results. So these kind of approaches ultimately they leads to all these approaches, they will result into hundred to thousand of molecules, and then we can screen those molecules against the particular targets. So and then we get a particular hit, and based on that hit, we some of the hits, 10 to 100 of the hit molecules, we will identify our lead molecule, and then we will optimize our lead molecule using the library, using the maybe like kind of virtual combinatorial chemistry, we can generate for the lead molecule, or we can design our own library of molecules and develop a structure activity relationship. And based on that structure activity relationship, then we move forward towards the lead optimization, and then ultimately uh, uh, the new drug candidate. Okay, so some of the filters which I mentioned, so it is not the so it's a very short period of time, like 1 hour. It is not possible to explain each and every aspect of the virtual screening method. So I'll just focus on some of the methods. So because each technique is like on its own, and it requires expertise of like I'll say like 1 hour each probably for each method. So we are just I'll try to uh, reduce this thing, and we'll use some of the most important parameters which as an academician, particularly during your research, which you can use. So one of them is like Lipinski rule of five, which says like the number of hydrogen bond donors should be less than the five whenever you are designing a drug molecule. Whenever you want to screen a library of molecules, so keep these things in mind. Like the hydrogen bond donor should be less than five, the molecular weight should be less than 500, log p of the molecule should be less than five, and the number of hydrogen bond acceptors should be less than 10. So this includes the sum of the nitrogen and oxygen atoms without the hydrogen attached, and this was the sum of the OH and NH groups which were there. So apart from that, like there are other rules which can be utilized. For example, if you are talking about like the drugs which we are aiming for the CNS as a CNS disorders, so what is the optimum log P for that? That also should be kept into mind. So approximately the log P for those kind of molecules is like 1.5 to 2.7. If that is the range, so you should always focus on like tomorrow you are working on a central nervous system disorders project, and you are saying the log P is 1. So should I proceed for that? So, so those kind of things should be kept in mind. Another thing is like one of the more interesting descriptor, or you can say the parameter, is like polar surface area. So the polar surface area of the drug molecule should be less than 90 angstrom square. So that these are some of the filters which you can use. Up now, the next part is when we are talking about that in the beginning, which I mentioned, that if we have only one more or more ligands which are available, so what kind of approach you will be going? So we will proceed with the similarity searching approach. So whenever we talk about the medicinal chemistry, this is the basic principle which has been laid down, that which says uh, that the similar molecules, they tend to have the similar biological activities, or if the molecules they are having the similar properties, that will be like they will be having the similar properties. So this is the example, like morphine, codeine, and heroin. They are like almost they have the similar properties, and as well as the similar structures. They are very chemically, they are very similar to each other. So in that way, if you want to uh, we assume, so they all of them they kind of belong to the narcotics drugs, so they can be used in one or another way. So but the basic assumption is that similar molecules, the similar principle, similarity of the molecule is resulting into the similar biological activity. So this is overall the we consider as a first step approach to assume that the for the ligand based virtual screening. Now, how the question is how will we calculate the similarity between two molecules? So this similarity can be calculated in different ways. One of the ways is like we can calculate the molecular descriptors. So we are assigning the different numerical values to the structures, where we like the different physicochemical properties, molecular weight, log P, molar refractivity, or polar surface areas. So we can calculate these properties, and we can compare two molecules based on these kind of properties. The another, which is more widely used in virtual screening methods, and also in kind of the artificial intelligence, it is like the two-dimensional properties. So what are those two-dimensional properties? They are like the fingerprints, then you have the topological indices, So, and we can use another approach, that is the maximum common substructure. So these are the some of the another approaches which can be used. And another approach, which probably many of you might be aware, that we calculate the different types of steric or the electronic fields of the molecules, which is being traditionally, which is being used in COMPA technique, comparative molecular field analysis. So either we can calculate some kind of molecular fields and we can compare, or we can generate some kind of 3D fingerprints and we can use them as the similarity searching. Now, when we calculate these different molecular descriptors, different 2D properties or 3D properties, now how you will compute their similarity between the two molecules? So we use a kind of a coefficient, which is like a similarity coefficient. Generally, it's a quantitative measure of similarity between two sets of molecular descriptors. So it gives you a quantitative measurement. Of course, you can you can simply calculate the differences, like if the molecular weight is same, so we can assume that they are similar. But this is a more exact estimate of giving us the similarity between the two sets of molecular descriptors. So we generate these different parameters and then we calculate the similarity coefficient, and then based on those coefficients we can assume that which are the similar molecules and should we take them into further steps of the drug discovery. So, so one of the which I gave you an example. So one of the parameters which is being used is like two-dimensional similarity search, where what we are doing, we are generating the fingerprints of for each of the molecules. So fingerprints is nothing but it's like kind of representation of the molecules in terms of the bits, which is your computer is reading the basically computer works on the binary unit. So it's the same principle that we compute some kind of the different bits for each of the molecule, and then we can compare that information using a coefficient, which is a similarity coefficient that is called the Tanimoto coefficient. So Tanimoto coefficient is very vastly used basically to calculate the similarity index between the two different molecules. So generally we assume like the chemical, if the Tanimoto coefficient values are more than 0.6 or 0.7, so we assume that two molecules are very similar to each other and then they can be used interchangeably with each other. Another method which is very much commonly used is the shape-based similarity. So this is one of the unique technique which has been developed by a company called Fred and sorry, Omega Scientific. So they have developed this a specialized algorithm where they calculate the similarity between the two molecules based on their shapes. So this is and they use a principle like rapid overlay of the chemical structure. So if you have the two molecules and if they fit with each other, and then calculate the Tanimoto coefficient or any other similarity index. So if they are having the similarities between each other, so we can say they are the similar. So similarity score is based on the common volume which they are sharing. So more and more this volume they will be sharing, so they will be like the more similar to each other. So this method is generally like considered as better than the 2D similarity search. So this is another example. So like we can see the ligand 1 and ligand 2, and they are like overlapped with each other, and like we assume the maximum volume overlap, and then we can see like how close they are. So this concept of similarity searching we also utilize in one of our project, and where we were utilizing this information for predicting the anti-drug resistance in different kind of biological targets. So the anti-drug resistance is a very common nowadays, where we are finding like mutations are making the different cells or different targets to show the resistance against different drugs. So for example, viruses, bacteria, protozoans, cancer cells, they are developing resistance to the existing drugs. So we have seen this is like a very challenging problem which has emerged not only in worldwide but like it explored widely in India too. Antibiotic resistance is becoming an alarming on alarming kind of a it's alarming all the nation basically where antibiotic resistance is being developed. So we have seen the drug resistance in the malaria. We have seen the drug resistance in tuberculosis. We have seen the drug resistance in HIV. We have seen the drug resistance in anti-cancer drug resistance. So the typical example is like triple negative breast cancer. So uh, we are not able to target those the triple negative cancer patients because there is a lack of drugs because no most of the targets which have been identified, most of them they are showing kind of the resistance at a very faster rate. One of the reasons of this drug resistance mechanisms, so they are like different mechanisms like that. It could be decreased transport to the site of action, increased elimination, metabolic inactivation of the molecules, ineffective activation of the prodrugs, and overexpression of the target proteins. So these are some of the drug resistance mechanisms. So but another reason which is developing some kind of drug resistance is because of the mutations in the therapeutic receptors or in the therapeutic proteins. So this is the case where we try to identify this method we have tried to apply it in kind of resistance against the HIV and particularly in the HIV protease. So why this HIV protease enzyme, which is like one of the main targets for the different drug molecules which are used in the treatment of AIDS. So one of the reasons is this receptor is very much prone to mutations, and lots of mutations have been identified. So we tried to identify those mutations, and we calculated the resistance of each of these molecules against these mutations. So this is overall the resistance of these molecules to the mutation. And uh, so the idea was behind this thing was that mutation should not significantly hurt the substrate binding or the protein's function in the virus. So this could be used as an advantage. So whatever the mutations are taking place in the HIV protease, but it is not disturbing the substrate or the substrate molecule, which is basically the different types of GAG and POL proteins which are of the cells of the human cells they are using, and uh, it cuts them at different places. So this was the idea that if we can uh, compute the shape of this substrate, and if we can we can calculate the volume which is occupied by this substrate, so can we use this as a tool to design new molecules, so as HIV protease inhibitors. So that's why we did this kind of an experiment, like the substrate and the ligands, they were overlaid with each other. So we calculated what is the total uh, volume which is overlapping with this and how much this ligand is lying out of the substrate. So this V out is was expected to correlate with the mutation resistance. So the theory which we laid down based on this, like, so the V out, if it is on the higher side, so we assume that more and more the ligands which we are designing, if we it's for them, if the V out is higher, so there are chances so it that the resistance will be on the lower side. So this was the idea. So we superposed the different substrates, and we calculated the substrate envelope density, and then we scored the different drug molecules. So these are like all the HIV protease inhibitors. We calculated their V out and V in and V total. We also like the known hydrogen atoms and the molecular weight, and we there was a like kind of an interesting thing that V out was like directly correlated with the resistance. So it was the correlation was 0.98. So which is like very significant value. So that laid down the concept of uh, this mutation resistance. This method we also tried to apply in other targets also. For example, one of the target was like ABL kinase, which is being used as anti-cancer target, and ATP was the substrate in that. Similarly, we have the chitinase, which is which has been used, dihydrofolate reductase in the malaria, neuraminidase, which is in the influenza. So these are the colored one. They are natural substrates, which which these different enzymes or the proteins they are uh, using them as a substrates. And basically most of these drugs they are basically the inhibitors of, they are in the competitive inhibition of these substrates. So that's the idea was which we calculated. So there also we found, like in ABL kinase, V out, it was correlated with the resistance. It was like 0.90. In chitinase also it was like 0.97. So these three targets we were able to meet out the criteria that the substrates and the inhibitors, if they are sharing their volumes or the shapes. So we can say we can compute the V out, and based on that we can say like higher is the volume which is lying outside the substrate, it's better for the ligands to have low resistance. We also calculated this thing with the different types of fingerprints. So we calculated the fingerprints of the substrate, we calculated the fingerprints of the molecules, different drug molecules, and we correlated this thing with the we correlated both the with the drug resistance, and that also was like kind of showed very good correlation. So in particularly in chitinase we had a 0.98 correlation, and in HIV protease it was 0.5. So there like 2D fingerprints we calculated, and it was working this principle. So this paper was like published in Chemical Biology and Drug Design. Now, so the second approach which is being used for the virtual screening approach is like pharmacophore modeling. So in pharmacophore modeling, the people we are very much aware as a medicinal chemist, like pharmacophore is the ensemble of steric and electronic features that is necessary to ensure the optimal supramolecular interactions with a specific biological structure and to trigger its biological response. So these are some of the key pharmacophoric features which are present in the any drug molecule, like the hydrophobic features, the aromatic rings, similarly like another hydrophobic features, or and a hydrogen bond donor or a hydrogen bond acceptor. So in this case, like it is we are representing in these two, but other features are like hydrogen bond donor, positive and negative features, customized features, inclusion, exclusion, volume spheres. So the basic concepts in the case of pharmacophore modeling or the pharmacophore searching is like if we have the number of ligands which are already known for a drug target, so we identify them. We try to identify the common features between them, and then we generate a kind of a hypothesis, common pharmacophore hypothesis, and then that common pharmacophore hypothesis can be used as a search in the databases which are available, and try to get the hit molecules, and then those hit molecules can be screened against the biological in the biological assays which we have developed. So this is the standard protocol which is being followed in the pharmacophore modeling, where we search the literature or the chemical databases. We try to find out some activity-based searches, and we try to identify some of the active and the inactive molecules. So it's better like whenever you do the pharmacophore modeling, we should also include some of the inactive molecules too, which can give you an exact idea like why there is a difference between the active and the inactive molecules. We do the pharmacophore modeling, we generate the hypothesis, and we search and in the databases, and we analyze the hits, and then we design the new molecules. We look out the synthetic feasibility with the medicinal chemistry inputs, and also we can go for the intellectual property landscape and the novelty searches, and then we proceed further for the synthesis of those molecules. So this is the typical pharmacophore modeling approach. We have a huge chemical space, and in this chemical space, which we utilize that collect the set of high affinity ligands for a particular target from the databases and the literature. So we try to identify the high affinity ligands or the highly active ligands. we try to generate their conformations, and then we identify the different types of features which are important, like the aromatic ring, hydrogen bond donor, hydrogen bond acceptor, electronegative group or electron withdrawing group, and hydrophobic group. So we find and align the common features, generate the common pharmacophore hypothesis, and then again search in the databases. So this is the general pharmacophore modeling approach which is being used. So this is the if you can see this the kind of a figure which I have developed. So this was like we did a work in the jubilant where we had a database of molecules which included like GPCR, ion channel receptors, and those other type kinases and other molecules. And then we generated the different types of molecular descriptors, and then we put them into a chemical space. So you can we try to identify like how the GPCR, how the kinases, and how the ion channel receptors, whether they are really lying in a kind of a similarity space. So at several places you can see like the green is highlighted. So this is like so from this we can pick one type of molecule and can be used for that particular type of a receptor. So similarly you can see the blue. So blue is like it's like kind of the molecules which are in one particular space for one particular types of receptors. So this kind of an approach, which using the pharmacophore modeling approach, we have utilized also to study the quantitative structure activity relationship models for glycogen synthase kinase 3 beta inhibitory activity of indirubin derivatives. So we generated a kind of a common pharmacophore hypothesis. We're using a kind of a training set, and then we tested this hypothesis in the using the test set molecules. So, but this was like kind of a paper which was we took the data from a particular paper which was published, and for the glycogen synthase kinase inhibitory activity of indirubin molecules. So we use that data for kind of this pharmacophore modeling based QSAR approach to develop that generation of that model. So we generated different conformations. We identify the different features, and then based on that common pharmacophore hypothesis, we develop and we try to find out which are the important the important features which are required for the molecules to have the GSK3 beta inhibitory activity. So that we have developed, and then we search this this hypothesis in the databases, the in-house databases of the jubilant, and then we use it further for the screening of those selected hit molecules. So, so these are the kind of searches which are available when we are doing the structure activity similarity searches approach. So now the another type of approach which can be used is basically structure activity relationship modeling. So in the structure activity relationship modeling, we use it to study whether the molecule is active or inactive. What are the reasons for that? So it's like kind of a classification model. So we use the knowledge of the known active and known inactive compounds to build a predictive model. So we are developing kind of a we choose a set of molecules which are active, as well as some of them they are active and some of them they are inactive molecules, and based on that we develop a predictive model and then use it to screen the databases. So we can use either quantitative structure activity relationship approaches, but in quantitative structure activity relationship approaches, whenever we start with this kind, so we need to get the activity data of the molecule. So we try to eliminate this part generally in the drug discovery, initially in the drug discovery. So we later on, but for the lead optimization, it is being used widely. So to basically for the screening purpose, or for the virtual screening of molecules, we use kind of a structure activity relationship model where we divide the molecules in the active and the inactive. So we try to identify. So exactly what we are doing. So we are having a training set where we have the known active and the inactive molecules, and we generate using the kind of a statistical approach like substructure analysis or recursive partitioning, support vector machines, k nearest neighbors, or the neural network. So these are the different types of clustering approaches which can be used, and they so kind of statistical different kinds of non-linear statistical principles which can be used, and then we generate a model of activity and we screen against the database, and we then score the molecules based on their top ranks, uh, and then we screen them the top rank molecules for the testing. So this is exactly the basic principle, the neural networks or the support vector machines, which is being used nowadays by most of the pharmaceutical companies in the artificial intelligence drug discovery methods. So which I will show you maybe in the next slides also. So this was the case study. So if initially we had a 50 active molecules and 12 inactive molecules, we calculated their polar surface area. So we separated them if they're less than 100, how many of them they are there. So out of the 100, only 5 molecules they were active, which were having the less than 100 polar surface area, 100 angstrom polar surface area. So only 5 of them they were active, and 9 of them the inactive molecules they were having this much of the polar surface area. Then we made another kind of a principle like dipole moment. So we calculated the dipole moment, and we also can use the log p values, and then we like out of those 5, the dipole moments were on the lower side, and five of them, and all the inactive molecules, their dipole moment was on the higher side. So the test compounds are dropped. So this is a kind of a clustering method, or kind of hierarchical modeling which can be used. So test compounds are dropped through the tree. Prediction depends on whether they fall into active or inactive ones. So that's what. So these all were like kind of the structure ligand-based drug design methods, similarity-based approaches, which can be used in screening of molecules as virtual screening tools. So the another method, once we have, suppose we have screened so many molecules or hit molecules, and then what we can use the molecular docking approach, which is a structure-based drug design approach where the structure of the target is available. So what exactly this docking is? Basically, docking is a kind of an algorithm for the determination of binding modes of ligands in the binding site. So most of the drugs, how they produce their mechanism of action is they are binding with the receptor through weak interactions, non-covalent intermolecular interactions. So like the hydrogen bonding, van der Waals forces, dipole induced dipole interactions, and the ion-ion interactions, hydrophobic forces. These are some of the known covalent intermolecular interactions. So using a computational algorithm, if we can compute this energies involved in the receptor and the ligand, so we can score the ligands, and then we can use this as an approach for the kind of a virtual screening approach, and this can be used to screen those biologically. We can screen those molecules which are the top scored ones, and which are showing exactly the uh, good interactions with the receptors. So that is the basic principle behind the molecular docking. So if we look at the molecular docking, basically, ultimately our aim is we have the three-dimensional structure of the ligands, we have the three-dimensional structure of the receptor. Using the docking principle, we dock the three-dimensional structure of the ligands into the three-dimensional structure of the receptor. That ligand receptor complex is generated, and we calculate the energy which is involved. So this is the typically a docking problem, how you can compute this thing and how you can score the ligands. So from where we are getting the 3D structure of the ligands, as I told you initially, like we use them from the experimental databases or the different types of libraries. So for example, you can use the library of National Cancer Institute NCI database, which is freely available. We have an SNH database which is freely available. That can also be used. nowadays, like probably you might have during the this corona pandemic, we have seen that most of our drug discovery is kind of going in the therapeutic repositioning approach, where uh, uh, we are not developing any new drug molecule, but we are trying the already developed or the known drug molecules for the treatment of this corona pandemic. So this is a new kind of an emerging area which has been being used in even in the cancers treatment also, and also CNS disorders too. So we can use the information from the drug bank. So this is the drug bank is basically the data set of all the FDA approved drugs which are available, and that can be used against your target, against your receptor, whichever is available, which you want, which against which you want to screen. So, and the three-dimensional structure of the receptors is available in the protein data bank. So that information we can collect from there. So in protein data bank, you have the 3D coordinates of the receptors or the proteins. They are basically generated using these two different techniques. One is the X-ray crystallography, and another one is the NMR spectroscopy. And then they are put into the PDB data bank, which is again free, and we can use this information. If by chance the three-dimensional structure of the receptor is not available, then we can use a homology modeling approach, where we can build the homology model. So using the another template, which is like kind of very much similar to the to our to our target which we want to use. So we can use the different tools like the Swiss model, modeler. These all are the different homology models making tools, and they are free. They can be used online also. So you can use that homology models as the three-dimensional structures, and then you can use that information for the doc. So whenever you are proceeding with the molecular docking, you should be careful what kind of tool you are what kind of docking tool you are selecting, what kind of principles that docking tool is going to use. So we generally have observed that this is our clear observation, and even our director in Jubilant, he used to say that docking is nothing but it's like you are putting a lot of garbage inside, and it's the garbage is coming out. Means kind of the lots of structures you are putting out, they will the docking tools, they will score them, and uh, uh, that information will later be of no use to you, and those molecules, they will never be active. So whenever you are doing that docking approach as a virtual screening tool, so uh, you have to be a little bit careful whether your molecules, whatever it is giving as hits, it is really making the interactions or those kind of things. You have to understand that is important. So whenever we start with the uh, kind of molecular docking, so the first thing is your selection of the protein which you have to take. So in the protein data bank, if you will check carefully, so for even a single target or the single protein structure you are taking, there are many protein data bank PDB structures are available, or the 3D coordinates are available. So which of them you have to select? So that you have to be careful. So these are some of the parameters which you can use. So you can use the ProCheck as an approach. This is a kind of an online tool which will tell you that if any residue is missing or not missing, so that thing can be used. So we also compute the resolution. So the lower is the resolution, lowest resolution protein you should always choose. That is kind of an approach. So then you identify the binding site. So in most of the cases, if the protein which you are selecting is bound with the ligand, so co-crystallized structure is available. So we already know the binding site. So that information we can use directly. But if in by chance if that protein is not containing the co-crystallized structure, so in that case you can use a tool like CASP, which can be used to identify the binding sites in the proteins. So, and then based on that, you can start with the docking. So once you have done that, you have identified the binding sites, then you will go for the proteins and the ligands preparation. So most of the uh, ligands you will be preparing using their, you will generate their conformations. So you will convert the two-dimensional structure to three-dimensional structures. you will add the hydrogens, and then you will compute the charges also, and then you will use that for as your ligand file as an input file. Similarly, in the proteins, so whenever you are starting with the protein, so we generally add all the proteins files, which are the PDB files which are available. They are not hydrogen added files; they are hydrogen depleted files. So we have to add the hydrogens in them. So that is the first step when you are doing the protein preparation, and then you have to add the charges, compute the charges, and then you will be doing the docking. Now, whenever we are going for the docking algorithm, two important things which you have to keep in mind. What is the search algorithm? So you know the docking basically is of divided into three different types. One of them is a rigid docking. Second one is a flexible docking. One is which has been recently, not recently, but it is also being used like induced fit docking. So rigid docking means we consider our ligands as the fixed. They are having the fixed geometry, and we dock them into the directly into the proteins. But in case of flexible docking, we assume our molecules they are flexible in nature, so they have different types of translational and rotational freedoms of motion. So keeping that into account, you have to use your search algorithm. So search algorithm, it will generate the different poses for the ligands which you want to dock. So there are different types of pose generation of the search algorithms, like the fragment-based approach, where you divide the molecule into small fragments and then you dock them in one by one. So then simulated annealing or the Monte Carlo approach, genetic algorithms, systematic search. So these are some of the search algorithms which can be used. And then once you have docked all the molecules, then you have to score them. So scoring is basically you have to give some kind of energy parameters to them, which you can tell like this molecule is binding better than the other ones. So for that we use another different types of scoring functions like molecular mechanics, empirical scoring methods, knowledge-based methods. So these are some of them which are so different docking tools. Whatever we are used, which are available for as a virtual screening tool. So they are based on the different types of scoring methods. It is like very hard to tell which scoring method is better and which kind of search algorithm is better. So it depends. Sometimes one of the search algorithm works, sometimes one scoring function works. So that depends. Some of the proteins like they are more hydrophobic. So with in that case, what kind of scoring method will be better? Some of the proteins they are more hydrophilic. So what kind of proteins, what kind of scoring methods will be better? So that all depends on the like kind of an experience or those kind of things. So this is one of the study which we as using the docking which we have done. Like we know like this was before this corona pandemic. Cancer was like one of the main disease which every country or everybody was focusing upon. So and lastly, like around 3 to 4 years back, it was a, like kind of a deep concern that most of the plasticizers and pollutants they are also kind of making some changes, and they are responsible for inducing different types of cancers. So plasticizers they are acting as the modulators in the breast cancer. So we try to analyze how these plasticizers basically, which are like different types of phthalates, how they are being how they are like kind of having an effect. So one of the student, this is a kind of a collaborative project with AMITY Institute of Allied Health Sciences. They have the Masters in Public Health. They are doing one of the project where they analyzed that some of the breast cancer, they collected the samples of the urine from the breast cancer patients, and in them they found like uh, most of the patients who were having the breast cancer, so they had lots of phthalates in them, and they tested it. They collected the urine sample and they did like LCMS studies, and based on that they found like those phthalates they were found as in their urine samples. So that was the idea we thought of docking some of these phthalates into the binding sites of progesterone receptors and androgen receptors and even HER2 receptors. So all of them we tried to dock them, and we found like some of them some of these phthalates, they are directly binding very closely with the natural substrates like estradiol and testosterone. So probably this is like we thought of giving giving a kind of an input that plasticizes. They can act basically as one of the reasons for the breast cancer. Another case study like which I wanted, which we have done is like we prepared the complexes of aceclofenac using with the beta cyclodextrins, and then how we computed uh, the docking of did the docking of aceclofenac into the two different structures. One was the monomeric and another one was the dimeric structures, and we computed the energy. So you can see very clearly that when it is docked into a dimeric structure of beta cyclodextrin, the energy is on the lower side. So this is more favorable kind of mm, cyclodextrin complex, which can be used for the development of the beta cyclodextrin complexes to increase the solubility of the aceclofenac. So over the years, so this is like I'll take another five, maybe 2-3 minutes only now left. So how the artificial intelligence, which is coming up as fourth industrial revolution technology, everybody is like moving towards artificial intelligence, where more and more of the data is being used as an input, and it is being processed through kind of the different types of supervised and unsupervised machine learning methods, and then using that information as for we process that information, and it gives us some knowledge or something. So huge uh, uh, the artificial intelligence tools are coming up. We can see like autonomous vehicles where we are focusing agriculture, space exploration, retail marketing, chatbots, Siri, Cortana, Google, Echo, all of them, they are what they are basically artificial intelligence. In healthcare and life sciences, we are wearing these smart watches. So that is also an artificial intelligence. So, we have the smart watches, which now like in the many of the companies they have launched, and they are saying like these smart watches can also measure your oxygen level, so that can also be measured. So that is also like one those all the things, they are basically the artificial intelligence applications which are coming up. So no sector probably is left uh, with the applications of the artificial intelligence. You can see Netflix, which everybody is using, HDFC Bank, and most of them, they have developed their own artificial intelligence tools. Like, so this is one of the agriculture company, Blue River. They have developed an artificial intelligence for irrigation, artificial intelligence kind of an irrigation system. Similarly, NASA, they have developed a kind of a rover which is being used for study of the Mars. Uh, so more and gaming industry has also emerged, the online gaming industry, which is like really the applications where the applications of artificial intelligence are being used. Artificial intelligence in healthcare, we can see it is being used in diagnosis, drug development, robotic surgeries. We know very well, like it's they are being used with the help of the robots. Cancer surgeries, eye surgeries, they are like very well known cases, very well known applications of the artificial intelligence. So mobile health, Internet of Medical Things, wearables, 3D printing. So these all are the different artificial intelligence in healthcare which is coming up. So it's like tough for me to explain. The time is less. So what exactly is artificial intelligence? So it's a process of using machine learning tool basically which predicts our the human brain. Basically, it's like behaving like a human brain. We so it's either a machine learning or a deep learning process. We use a natural language processing, which is a kind of a computer language. So maybe we can develop a Python code, we can develop a C++ code, so build the artificial intelligence models and they can solve the complex problems. So it's almost the same thing. So we basically artificial intelligence is like we collect the data, we store the data, we process the data, and we generate the information and then use it for the purpose which we want. So how this information is being used in the drug discovery. So we have the nowadays like most of the there is a tremendous change in this application of drug discovery. Last couple of 2, 3, 4 years, you can see more. I'll show you in the end. This is the thing. Like more than 2,000 companies they have come up and they are giving the AI applications. They have developed they developed their artificial intelligence platforms and they are selling to the many of the pharmaceutical companies and for screening of different molecules. So they what exactly they are using. So they have the enormous number of chemical spaces there. They pre-screen the molecules, do the similarity search, pharmacophore search, docking, and then we get out the hit list post screening like clustering and hit evaluation, and we generate the new lead that is identified. So this process is like very fast. So even during the this corona pandemic also, like one of the companies they developed some of the molecules and they said they developed like within a month they came out with some highly active molecules. So of course, so later on the things are like they have to be going for lead modifications and those kind of lead modifications, lead optimizations and different types of clinical and pre-clinical studies. So all those things are different and they are time consuming. But at this step, they came out within a month. They have developed some of the hit molecules. So this is overall a artificial based data mining. We have a compound database. We extract the chemical features based on their fingerprints, and then we just based on some kind of machine learning methods like instant based learning or model based learning methods, we generate the different models and then we can use them for a kind of an AI based tool for the drug discovery. This is kind of an overall flow how the artificial intelligence is being used in drug discovery. So and these are some of the companies which I was mentioning, which have come up in the last 5 to 10 years. So this is a huge number, more than 2,000 companies. They are now giving their artificial intelligence models to and helping the drug discovery in a faster way for the different pharmaceutical companies. So that's it. I think almost the time I have taken is more. So thank you so much. Again, I would like to thank the entire DIPSAR family and DIPSAR family for giving me the opportunity to share my ideas and my research experience over the years. So thank you so much. So if you have any queries, so you can mail me on these two email IDs. Yeah. Okay.
Very nice lecture, in a very tipsy way. You have tried to give the overview of the total drug design process.
You are not audible clearly.
Thank you, Professor Vinay. You have given a very tipsy idea about the drug design, computer-aided drug designing, molecular modeling. Okay, and in short, the latest field which is emerging here, that is the artificial intelligence in the drug discovery process and its importance. So in future, I think it will be wonderful topic to be covered by you in future for our research scholars as a faculty member, that is especially artificial intelligence and various software.
Sure. Thank you so much. Thank you for giving me the opportunity.
Your valuable time, and you are always supporting us with your ideas and your research activities. So I wish all the best for the day, and thank you very much. Thank you, madam, please.
The session is open for question and answers. The participants can either unmute themselves and ask, or they can put it in the chat box. So the question first has come from Dr.
So what I was thinking, ma'am, if we can take like a couple of questions only, so because another speaker of yours, he has joined.
Okay, fine.
So then maybe like he's waiting also. So
Okay,
it's better like if
let us take just 3 questions and then we can.
and so one of the most important part which probably he will also be sharing, like how once you have docked the molecules using your molecular docking approach, so what are the next steps which you should look into to what kind of different computational things we can use. So that he will be talking like molecular dynamics. That is another principle which is very important. So
Should
he'll
we
be
do
sharing on
something
that.
like
So
this?
yeah. So
That both the question answers we can have after his
Yeah,
lecture.
that will be better. That will be
yeah, yeah, sure. If it is possible, then it is really good.
We'll ask.
If all the participants they agree for this.
Yeah, they will
Sir will
agree.
be always there with
Okay.
We
us.
understand,
Yeah.
they're so cooperative and nice people. Uh,
Yeah,
I
no, that's
request,
fine.
I request all the delegates to keep their questions written in their diaries. And after the second speaker, who is probably guru of Dr. Vinay Latha, is after his speech, we can ask both of them together. So I request all the audience, we will take the questions later.
Wait.
Please unmute yourself. Madam, please unmute yourself. Yeah.
Hello.
Am I audible, sir?
Yeah.
Yeah. So let us move ahead with the second speaker of the day. Actually, I have to give certificate to Dr. Vinay Lathar. Should we finish that first, or Dr. Vinay Lathar would be available later?
Yeah, we can finish it with the help of his mentor. We can finish.
So, though we
yeah, I'll be available. Yes, I'll be there.
available, so should we do the honors together, sir?
Also,
Sorry, ma'am.
Should we present the certificate to you as well as your guide at the end of his speech?
I'm not able to understand actually.
no problem.
Okay.
Okay.
Sir, let us, Bakuri sir, I request you to honor the first speaker with the certificate. Though we normally honor with a bouquet and certificate and token of love, this time only it is a certificate. I would be displaying the certificate.
Sure, ma'am. Sure. Okay. So it's like I welcome Vish. So we have worked together for around 3 years. During my stay in Portugal, and we did few projects together also. So few of the publications also we have come together.
Congratulations.
So he has like a huge experience, in the basically he's a core computational chemist. So I am an kind of an application scientist, So I'm using the different tools, but he has developed different tools basically. He has developed his kind of a docking tools on his own. He has like a vast experience in molecular dynamics,
So he
how
has
those things
used
are being
AI in
done.
developing the tools.
Yes, so
Great.
he's a hardcore, computational chemist.
Great.
Vast experience in quantum chemical calculations and those kind of things, molecular mechanics, and so I am the application scientist part of.
We are lucky to have the Guru
Yeah.
Shishya Jodi together.
Yeah,
And,
sure.
it's always a pleasure that the student is paying his tribute to his guru or guide. So it's something great. Everybody can learn that after learning, even after 15 years. Dr. Vinay has a great, what do you say, respect for his guide. And
Yeah, I really admire him. Yes. I really
Thank
admire
you
him.
so much, Dr. Vinay, for bringing your guide to us, introducing him. So our next speaker is Dr. Viswal Das Kairias. He is he's a senior researcher at uh, Institute of Biotechnology, uh, Vilnius University. Before that, he was a postdoctoral research associate at the University of Madeira, Funchal, Portugal. From 1999 to 2005, he was a postdoctoral research associate at Center of Advanced Research in Biotechnology at Rockville, USA. In 2002, he was a research scientist at Essential Therapeutics, USA. And before that, from 1992 to 1997, he was a PhD student as well as a teaching assistant at University of Hawaii, Honolulu, USA. He was a research assistant at Vilnius University from 1989 to 1992. He is PhD in chemistry from Vilnius University, Department of Chemistry, Vilnius. He also has a diploma in chemistry, and he is such a humble professor. He has not given any information about the awards and publications, so I do not have idea about it. I request Dr. Viswal Das to appraise us about his achievements, about his field of research, as well as his awards and publications.
Okay. Okay. Let me. Okay. So, do you hear me well? Yeah.
Yeah.
Yes,
So,
please. We can hear you.
okay. Yeah. So I have about 50 publications, and I didn't include them because they requested short CV, and I don't have many awards. I can say, but I have some experience under my belt. And the previous talker, Vinny, gave us very thorough presentation about the field, and this one will be a little bit lighthearted. As you can see, I am in my daughter's room for my family, because I'm stuck here because due to the COVID in Madeira, and you can see. So this is my daughter's room. So my presentation will be less more of my personal first-hand experience in certain things which could be very useful, and I try to be educational and give you tools which you may need to perform certain tasks. Let me see the start screen sharing. Oh. Okay, so how do I switch on the presentation now?
You have to go to share screen.
You can share your screen.
Oh, okay. So, okay, now I think I'm allowed. Yeah, okay. Let's see. Oh, okay. Okay. Now you see, okay, wait a second. Okay. So I'll be talking about exploring biomolecules using molecular dynamics, but I will touch upon several other topics briefly. And you can see in the end, I put my simplified email, visvaldaskairis@gmail.com. Did you know that the dot is arbitrary? You can put any dots you want in Gmail. Just a little trivia. So I'll start a little bit about my university, because I'm probably okay. Let me see. Okay. Let me ask you. You see well, for example, when I move this, you see the present slides well? Yes. Or it's not obstructed, no, because I see like the people the video is obstructing a little bit this, but I think you probably are okay. So I'm from Lithuania, and I'm from Vilnius University. And Vilnius University was founded in 1579, and it was one of the first universities in Eastern Europe. But I won't talk much about the old university. It's very beautiful and so on. But the place where I'm working at is Life Sciences Center, and which was built very recently in 2016, with partly European Union support, and it won several architectural awards. I'm not sure why. It's just the glass ant hill, and you can check. This is a long address, but you can check virtual tour and see how it looks inside. You can Google Life Science Center Vilnius University virtual tour, something if you want, of course. So what else? A little bit about, not many people probably know about my country. So Lithuania is a small country on the shore of the Baltic Sea. And it's part of European Union since 2004 and joined NATO in the same year. And joined Eurozone. We have euros since 2015, and about 3 million people. And religion is predominantly Catholic. As you can see, it's far away from India, right? Okay, but however, there are some ancient India connections. I couldn't resist this. So it's Lithuanian language is part of the Baltic branch of Indo-European languages, and presently there are just two living languages of that branch, is Lithuanian and Latvian. Latvians are our neighbors to the north, and also there is one extinct language is Prussian, or some people say is old Prussian. So so what is the connection with India? So it's Lithuanian language is considered to be the closest to Sanskrit from the living languages. And therefore, there are many word similarities. For example, sunus is son, avis is sheep, padas is foot. Viras is man, dumas is smoke. And I was, several years ago, was shocked actually learned that in Latvian, Brahmanis means priest. Okay, and then another quaint connection is our largest river, Nemunas, is some say is related to Yamuna River, which is holy in India, and somehow this refers to the river which flowed through the prehistoric Indo-European homeland, and nobody really knows where it was. I think maybe you have some better idea. Okay, now we go to the scientific part now. Now we'll talk about a little bit about docking. Actually, as Vinny mentioned, I did had the hand in writing some docking program, but I stopped using it right now, and I will tell you in a minute what I use now. So sorry, this Vinny already explained. This basically docking problem is lock and key concept. There is a target and there's a ligand, and ligand is supposed to fit the target. And the docking problem is, why is it a problem? Because there are many possible combinations of degrees of freedom in space, and the correct structure is just one. So out of many conformations, the program has to choose the best. And and to make calculations faster, they use molecular mechanics or force fields as opposed to quantum mechanics, which is supposed to be more accurate method. And actually, molecular mechanics parameters are normally derived from the quantum mechanics. So basically, there is these terms which refer to the bond stretching, angle bending, dihedral angular rotation, improper dihedral bending, van der Waals interactions, and electrostatic interaction, which is probably the most complicated term in reality. So binding energy calculation is a very calculating binding energy is a very difficult challenge, and there are and to follow what is newest in the field, you would like to read the papers of Sample, D3R, and Similar Global Challenges for the latest trends. And also, if you have your own great scoring function or method of docking, you can always participate in those competitions. And try to see if your algorithm is doing well, how your algorithm is doing well. And actually is how to say the self-promotion or something. So I wrote a little chapter in one review article, and so I covered. I did quite a bit of search and looked for the best ways to calculate binding energies, and I'll mention some of the methods here in this presentation. And if you want, you can drop me a line. I can send you the PDF because it's not free. So now about the docking. So in my experience, my recent personal discovery is SMINA program, and it is an autodock fork with additional with some additional bells and whistles, but it runs from the command line. In other words, it's just for Linux or Unix-like system like Macintosh. And one reason why it is very good is because it can use Vinardo scoring function, and it's one of the best in the industry. Its Vinardo scoring function is really great. And for other scoring functions, in addition to Vinardo, I also recommend Convex PL by Grudinin, and I know it a little bit because Grudinin is collaborating with our bioinformatics group, and the performance of this Convex PL is similar to Vinardo. It's comparable to Vinardo, but sometimes it's useful to use several scoring functions because, for example, these two, they're based on completely different principles. For example, Convex PL is artificial intelligence based scoring function. Okay, now we go to molecular dynamics. So I won't go into how it's done and everything. I just probably put some pretty pictures. And so molecular dynamics models realistically the systems, and which it must include solvent molecules. There are some also some methods where they use implicit solvent, but I somehow don't believe it. It's much faster, but I don't believe somehow in it. So as you can see that this is basically some cell which is filled with the molecule in the middle and filled with solvent molecules. As you can see, there is lots and lots of interactions to take into account. So therefore, it's very demanding method. And something which I encountered not by how to say, not consciously, but sometimes by sort of by request. Some people request, oh, can you calculate something, and then very often is some mixtures of solvents. And it turns out that mixtures of solvents can give interesting and useful information. For example, if you can see so this is the solvent with waters removed. So what you see is protein and acetone. And here is the same, but water removed, but just third butanol. So, and you can see that there is a very, very strikingly different distribution of solvent molecules on the surface of the protein. It's really amazing. For example, here you see it's completely covered. So and actually it can be used to probe hydrophilicity or hydrophobicity of the binding sites, and there is methods for that. And this is put some reference to that. Just very briefly about mixed solvents, and actually I'm doing some calculation of mixed solvents right now. So I'm also we'll mention free energy calculations. It's a kind of strange name because free energy. Okay, so binding energy is all binding energy is free energy, but there is a special name for that. It's free energy calculation. So what they mean is actually sometimes it says alchemical calculations. So what is happening? So a molecule is mutated, for example, here there is, I assume there is probably in this places there is hydrogen is mutated into methyl or something. It seems so. So basically, it's alchemical because it's mutated. So, but how to say? You cannot change it quickly because you get like big jump in energy. So is the proper way to do it is to gradually emerge methyl out of hydrogen. So gradually evolve, and like this, they calculate some kind of hysteresis energy and get much more accurate energies. Although there are some studies which where it says it's actually less useful than it seems, and so on. But anyway, but I don't go into those details. And in that review paper, I think I mentioned something about that. Okay, so now what if you are short on resources? So you could try coarse-grained molecular dynamics. So coarse-grained molecular dynamics is basically molecular dynamics where several atoms are replaced by one. So for example, in this Martini force field, which I like to use, you can see 4 or 5 atoms are replaced by one particle. So then you can see that this is how it is, this replacement schemes. And what is also important, that 4 waters are replaced with one particle. So which means you can imagine how much fewer particles you need to simulate. Okay, so this is an example of something which is, I think, it's not very properly physically done, but I did some calculation on this. But I think now there is some mistake in these calculations in the setup, in the whole setup, because it's half membrane. So it's connexin is a membrane protein. As you can see, membrane. There is a very nice Martini people. They're made some very nice scripts to build their systems and to make so that other people can use their program. So you can see there is a membrane and there is a solvent and there is a protein, and this is how the system looks like. And I'll show you the simulation. So this is if you put hexanol into this system, so you can see where it goes, right? So hexanol it probably will be one hexanol molecule will be 2 particles. So okay, so let's see when it finishes. It goes. Okay, so you can see it's kind of distributed here and there and everywhere, right? And what's important, as I said, if you're short on resources, so this was done during maybe 1 day or maybe a couple of days on 4 processor PC. It was probably 5 years ago. So you can see it's really good on resources. And this is when you have non-anol, which is 3 particles, and you can see this behavior is radically different. You're gonna see that it coalesces into drops, and it's very satisfying to see this happening. Okay, I think that's very interesting. It's not published. There's many things which I did here. Most of the things that we did is not published or in the process, works in the process. And sometimes things which are not published is because very often there is one point, for example, they have data and one point is off, and then I don't know why it is off, and then I cannot publish. That's it. All right, and okay, now another thing is non-equilibrium molecular dynamics. Non-equilibrium molecular dynamics, this means that it's to model systems which are out of equilibrium. So in other words, you force the system to do something. So I really highly recommend Plumed, the plugin to perform non-equilibrium molecular dynamics. Normally I use GROMACS, but it can be attached to any well, almost most of the molecular dynamics programs. And I will be talking a little bit and mentioning some of those tools. Okay, so so the first thing, the most probably useful thing and the sort of easiest to understand is tiered molecular dynamics. So so basically what happens, you can, for example, where you can force some variable to change, and variable normally is, for example, the distance between one molecule and another molecule. And when it moves, the accompanying work is measured. So the most popular variable, as I said, is distance between the atom groups or separation of groups molecules, but it could be anything. And the Plumed has scores and scores of different collective variables. For example, RMSD from reference structure. For example, you can force protein from one conformation to another conformation and measure the work which is required to do that. Number of hydrogen bonds, it can count it. Number of contacts, dipole moment, angles and torsions, and so on. So in other words, you can twist angles. You can do anything with a molecule, with the protein. Well, molecule. Yeah, protein is also molecules. So this is one work which only part of it was published, but this part wasn't published because there is some points which fell off of the curve. So so DNA clamping opening. So clamping proteins. So what happens actually? DNA goes in the middle of this ring. So it's sort of something which is very curious about this protein, which my boss is Czesław Wenslowas is a really master of homology modeling, and he's a winner of many competitions, international competitions and world competitions for homology modeling. So something which is curious. So these organisms, they have very different sequence, but they all have clamp rings, but the sequence is very different, but the shape is the same. So this is an example where the protein function takes precedence over the sequence. So for example, if you take like bacterial and for example, in human, the clamp rings, the sequence similarity is, for example, 15%. So it's really not much. So basically, what's happening when you do stir dynamics? It moves. You move. It opens this one. It opens the ring, and the half time of the opening is calculated here. So this is 4 hours. Of course, you cannot model 4 hours in real time because it will take years and years of simulation. So we need to pull computationally. So fragments are pulled at constant speed, and this is measured using the Jarzynski calculation. And you don't have to worry about it because Plumed reports it automatically. And well, the one principle is that you have to pull kind of slow enough. If you pull too fast, then this the energy gets exaggerated. So here's when you pull, you get sort of profile of energy profiles of pulling. So this is one of the data to get. So this is something picture of what you would get, and some of them doesn't correspond to the experimental data. That's why I didn't publish this. Okay, so now as I said, you can force protein into anything. So this is my little experiment. So this is insulin and a calculation when I used ratio between alpha spirals and beta sheets as a variable, and try to force that variable from one end to another. So so basically it has lots of spirals. Insulin has a lot of spirals, and beta sheets is formed, if you know, when it forms plaques, right? So I try to model this, and then as you can see that this is from spirals, you get plaques. So basically, but another lesson from this, you can force protein into anything, do anything. You can squeeze, stretch anything, but the thing is, you have to think really well. Does it really correspond to reality? You know, you can do anything. So sometimes I see in some people send to review some papers, and then the people do some crazy things, and then I say I have to think. You have to say also to them, I know you can dock anything into anything, but does it mean you get some energy? Does it mean anything? Now, so you really have to look at the experiment. Experiment is everything. And this what we do is to try to help. That is my little lesson here. Okay, so so it's another thing which I did, steered dynamics, and actually I'm doing right now as we speak, is collaboration with University of Madeira, is binding between doxorubicin and dendrimer. So I sort of developed my own little protocol. So I take samples, so put randomly ligand or doxorubicin, so you can call host and guest. So I put the guests randomly and then pull it in, wait a little bit, and then pull it out, and then write here the binding energy, binding affinity. And you can see that that this is really nice, but we get 1 point off. So now I'm trying to redo something using this mixed solvent now. So it's always, there is 1 point off. Always this is a problem, and that which makes it very hard to sell. All right. Okay, so now another one is metadynamics. I was this is very interesting method, which superficially looks like steered molecular dynamics, which goes back and forth multiple times. So because if you steer dynamics, you steer, for example, from 0 to for example 15 angstrom to separate. But in metadynamics, you go back and you back, and then you again pull away, and then again go back and go like this. But you don't do just like this. But what you do at the same time, you pour some potential, depending on where you stop. You put some little piece of potential there. So, and like this, they call it artificial sand. So basically, so this is our shape of free energy curve. So basically, gradually this fills up with the sand. So eventually you get this equal potential. So in other words, it becomes flat potential. So what you do, just this is bias. So basically you invert that, and then like this you just count how many potentials you put, and like this you get the shape of the curve. And like this metadynamics allows to use, for example, if you have 2 potentials in Plume, you can do that, and I tried that. You can get potential energy surface. Yeah, but recently, actually, not some years ago, I discovered that there is some little bit of sort of bug related to the periodic boundary conditions with this method, but I wasn't able to resolve it actually. And I'm kind of a little bit stuck with this. So I put it into drawer, this idea, how to resolve this. All right. Okay, so now about the analysis of MD conformations. So one tool which I really recommend is Plumed, is for making non-equilibrium conformations. Let me see the time. Okay. So is this one, is Bio3D. I was very happy when I discovered it. So it's by Professor Grant. And it's implemented as a package in R. So we have to learn a little bit about R in the process. Bio 3D. So so I'll go into applications about Bio 3D after I introduce a little bit about CRISPR protein. So this a little bit off topic, I guess, because CRISPR is not really like well, it's very big. There is big brouhaha related to it, but it's it's not really to do with the small molecules. It's something about behavior of the protein. So it's CRISPR associated system is one of the mechanisms that prokaryotes defend against viruses, and this very hot topic. Articles appear in top journals, and it's used it's already used in gene editing, and in mammals, and it can be in other organisms. And the way it works is, I was in this is the defense of this lady who has a very nice presentation in Science. So basically, when virus attacks bacterium, then the piece of RNA is included into the CRISPR array, and then what happens is the bacteria creates effector complex, and the next time virus attacks, it cuts that piece of virus RNA. So okay, this is like kind of empty slide almost, because I just wanted to show that there is CRISPR proteins. There is immense number of CRISPR systems, and there is like they divided into two classes. And I want to tell a little story because I think it's can be useful and interesting. So this guy in the center is from our university and actually works in our joint life sciences center and even our institute, because I'm part of Institute of Biotechnology. And recently a few years ago already, he got some prestigious Kavli Prize, which is from Norway, and it's pretty substantial. And also got the prize got two ladies, Emmanuelle Charpentier and Jennifer Doudna. And another guy who co-discoverer of CRISPR didn't receive it. So so this guy now, Virginijus Širkšnys, is the most arguably is the best scientist we have in Lithuania, so could say, and possible maybe even for Nobel Prize, I don't know, nomination sometime in future. I'm not sure about that, but I think what is interesting is a little story about this discovery. The thing is, he discovered it first, and then he sent to some journal. I forgot what the name of the journal, and it was rejected. So he sent to another, I think he sent to PNAS. And in PNAS, they said, okay, so you have to do this, and you have to do this. And then he was writing back and forth. And during that time, one of these, I forgot actually who it was, one of these ladies, they sent a fast track in the Science or some similar journal on a fast track, so without much check, and got first. So it's like this, we would have been first, but then we're not first anymore, and so on. And then there is also a very bitter feud between Jennifer Doudna and this Feng Zhang with patent, in terms of CRISPR patents, and we are not able to participate in those patent fights because we don't have money. But this to show that being small guy is sometimes it's not very good. And you can see that. Well, anyway, you can read about this in there's some news in the internet. So anyway, so I will be telling about what I was doing to the CARF proteins. So there is one CRISPR system, proteins in CRISPR system have CARF domain, and there is CSM6 and CSX1. Okay, and why they asked us to look at it? Okay, so what the thing is that is what is happening is here ligand attaches to this part, and in this part cuts the DNA or RNA. And the big mystery is how the signal gets transferred from here to here. That's called allosteric action. And it's can be 100 angstrom different distance. And actually, they have even structures with ligand and without ligand. You don't see much difference. So it's really a big mystery. So and this, it takes me already, I don't know, 4 years. I'm doing this calculation. I'm still doing. I have some clues, but it turns out it's more complicated than I thought. And also one thing about this CARF domain. CARF domain is basically can be CRISP associated and not CRISP associated. So it means that has other function, but the ones which are with CRISP associated, so they are something to do with the cutting here. Yeah. So I did the molecular dynamic simulation. It's 200 nanosecond MD simulation, which is quite a bit for such a big protein. And I did without ligand, with ligand, and there's also with the deoxy version of the ligand, because it's also not inactive. So without ligand and with the deoxy ligand, deoxy adenine, cyclodeoxyadenine should behave similarly. So for the analysis tools, but I'm more, I think I would like to be more educational, because I don't have many things to show to something like great results or something, but I want to show you the tools. So something which I found very, very useful is morphing tool, which is little tool, which is not very, very visible, but it's in Chimera program. So morphing, so basically you show, for example, one conformation, another conformation, and it creates for you, it morphs one into another one. So like this, you can see immediately what is happening, where is the biggest changes and where the smallest changes, what side chains rotate, what and so on and so on. And another tool is with Bio3D. Remember I told about Bio3D? So it's a principal component analysis. So principal component analysis, basically what it takes all the coordinates of all the snapshots of molecular dynamics and superposes them, and then tries to get the most characteristic movement. That's why it's useful, because it's you can see all the movement in one at a glance. And another one is community network analysis, which I'll talk to you a bit later. I'll show you a little bit later. Community network analysis. So these three tools, I found very useful in this particular problem. So so first thing is this is principal component 1. So it means the main movement of some of the proteins, but these proteins are not the ones which I'm looking for. This is another CARF domains, because there are some of them there. Remember I told this, not even CRISPR related, but they have the same domain, CARF domain, and you can see there is some flapping movement like a bird, right, or scissors. So we might expect that there is something to do with this flapping. So now another, they talk a little about the atomic motion correlation analysis. So basically what it does, so it creates dynamic correlation matrix. So it calculates this B of 3D. It takes, okay, so atom, for example, in residue 1 and residue 324, how correlated is their movement? How correlated is their movement? So for example, if it goes out of phase, goes out of phase, or going to be in the phase, or it could be very far, right? It could be close, but it calculates how correlated is the movement. So it builds the correlation matrix of the movement. So you can see there is a plus correlation, positive correlation, and negative correlation, which is purple here. Actually, yeah, you can see that there is a scale here. So, and then based on these correlations, they build a tree. You can always, when you have correlation matrix, you can always build a tree. And then based on this tree, they cut the branches which are most characteristic and into communities. So like this is called community. So basically what it means that atoms in a community, they move together. Can be out of phase or in phase. Okay, so but they are correlated. And by themselves, these communities also connected with by some sort of correlation. So and the size of that correlation is reflected in thickness of this stick. Okay, so it's very useful little tool. Okay, but I always think I was very excited about this CNA analysis, but always think, okay, movement is the first. This is movement is the fire. This is the smoke. Okay, so I always try to think of that the movement is the first. So it's very pretty, but you have to think about the nature. Okay, so and there's another one example from high profile paper is something like with about kinases, and you can see the different communities and how they are correlated. This is also a very nice picture for putting in a paper with the correlation. And then let's going back to those to our carved proteins. So you can see that without the ligand is you get this movement here, and with the ligand, this is the ligand. I don't draw ligand here, but is here. So there is no movement, no flapping movement, and you can see also it reflects in the communities. Okay, so and then how this can be interpreted? Okay, so if there is no ligand, it means this DNA or RNA can go inside here and then get out. It's inside, but here when you have ligand, it goes inside and gets locked. So basically, it's locking. Okay. All right. So now, but the problem is reproducibility. So I get what I did the 5 different runs, and then in some of them it moves, in some of them it doesn't. So now I'm doing what this is. So what to do if you have reproducibility problem? And it turns out I looked at it a little bit, and it really depends on the backbone. In one place, the backbone angles they have very big influence on the rest of the protein. So we really need to sample faster. And how you to do that? So what I'm looking right now is replica exchange molecular dynamics, are REMD, also known as parallel tempering. So also implemented in Bloom. This is a schematic. This is a schematic of this. So basically what is transfer, but it has to be run in parallel. So for example, if you have 4 processors, so you run one copy as 300, another copy of 400, another 500, another 600. And then every, for example, 100 steps, 2 neighboring copies, they can be swapped depending on the energy. So it's some kind of Monte Carlo mechanism. And like this, if you run it long enough, so basically very hot protein with much easier to overcome barriers can trickle into lower temperature, but this is into room temperature conformation. So in other words, this protein, hot protein, can actually cool down and go into the 300 degree conformation. So like this, you can get the conformational changes much faster, much faster. Converges to global minimum much faster. Okay. So actually I'm finished now. So I'd like to give thanks to our Department of Bioinformatics. You can see this very stern male company. I don't know why girls, they're afraid of this computations, I guess. Also, I must also say thanks about the Department of Biothermodynamics and Drug Design and Dr. Domantas Matulis. Where is he? Oh, one of them is actually, there's so many. There's very big group, but they give me a lot of work. For example, yesterday they sent me, do you know how to explain this data? And then, oh my God, they have so much work. I don't know how I'm gonna do that. It has to do always to do with docking, actually. Have to do docking. And then what they looking mostly is carbonic anhydrases, which has zinc, which is a challenge, and the HSP protein. if you have some ideas about those proteins or something, you can contact me. And also, I would like to thank Dr. Rodrigues and Helena Tomas, here we spend in Madeira, and also to Vinny and Dipti. So this is the picture from Madeira when they were there as a postdocs. So this is my short talk, I guess. Yeah, we'll be happy to take any questions, or you can write me an email, drop me a line.
Hi, Bees. Thank you so much for the talk, and thanks for accepting the invitation. It's so nice to see you virtually today.
It's my pleasure.
It was a very informative talk, and it has actually completed what Vinny started. You have ended. So the session is open for the questions. Now the participants can ask questions from the first speakers, from the first and the second speakers both.
I think there was a question in the chat session of the first speaker. Deepti ma'am, can we open that and see the question? Because I'm unable to check from the first lecture.
Actually, ma'am, same is case with me. My chat box has, okay. We have a question from Sapan Shah. Sapan Shah, you can start your video.
Yeah, hello, good afternoon, sir.
Yeah, we are able to hear you. Please go ahead.
Sir, I came across one CAPFLEX 2.0. This is an online molecular simulation software for the proteins. So it just required to upload the protein structure or the docked protein structure along with the ligand. So how is this? Can it be useful? Because a very few research paper has reported in their references. CAPFLEX 2.0.
Well, personally, I don't know nothing about. I'm sorry about this method.
So, what exactly is your purpose, Sapan? Can we ask,
Sir, this is for
maybe based on that we can suggest?
So this is for the molecular dynamics they have used, or for
Okay.
studying the rigidity of the protein structure. So
So if you are very much familiar, if so some of the best molecular dynamic tools, and they're available free also. One of them is like Amber, if you can use. So that is
yes.
one of them, and Gromex is another one.
Yes.
Which you can use. So
but that require hardcore processors for the Gromex or Amber. But this is online. We just have to upload the protein structure or the docked protein structure. So
okay. So
it does
you
not
can try
require
it. It's not like basically it gives you an analysis how when you are taking a dog protein and then you are running some molecular dynamics calculations on that, so you can see how the changes takes place in the residues basically when the ligand is bound with the receptor. So those you can run it and you can see the changes. So it's not like that it is of no use or something like that. We cannot say any. This is like, so you can use anyone. It's not an issue, right?
So second is you said in your first lecture it is very difficult to select which process, which method we should go for the docking.
Yeah. Please.
So as a learner, if one has to start, so what you recommend? Because I come
See,
across.
yeah. So that is very much clear. So the things is, it depends on the kind of resources we have. So most of the paid software, they are so expensive that most of our academic institutes probably cannot afford them. Like being in a kind of a private sector, or we understand the situations there. So it's better like if you can use some of the free software. So Autodoc is like one of the recommended one. People are using it, and they are getting some. In some cases, they are getting good results also. So, but if you have access to, even you can use the Fred also. They can, you can have an agreement with the Omega scientific, which they are developing that tool. So you can use the Fred also. They for academics, they kind of sign an agreement for 2 years. It is free, so you can use that also. So, but
Omega.
like, so most of the cases, what our observations are like, probably I would recommend, if you have an access to Glide or something from Schrodinger.
Huh.
So it is like one of the best
Okay, sir.
at this moment. So that is one.
Sir,
Uh,
sir,
yeah.
there is
So,
a
and the other thing is, so don't totally depend on the docking
yeah,
results of which you are producing. So it's better, like after that you run some molecular dynamics simulations
correct.
also. So it will give you a more better confirmation of the ligand in the protein.
Yes, sir. Yes, sir.
Yeah.
And one advanced feature of Autodoc Vina, one recent paper in 2020 is published on Firefox algorithm Vina, FWA Vina.
Yes, yes, yes. It has also come, and there is recently we also have an access. Somebody has written a kind of a script, and that script is like in a GUI kind of a mod. So you can use that also. So that is like
So
you
what is the
store.
name?
I'll share with you if you can. You can send me an email to
Okay,
me,
sir.
right?
Okay,
So
thank you.
I'll share
Thank
it.
you.
So
Thank
just you
you.
have to give the two different locations of the folders where your protein is and where your ligands are there, and then it automatically does the docking. Otherwise, like Autodoc is a kind of a time-consuming process. So, and many times, it kind of gives the Autodoc also, it is not producing very good results what we expect.
Hmm.
So that we have compared, like during this SARS-CoV-2 studies, we also have done some calculations using Autodoc as well as Glide. So we saw some comparisons also where Autodoc was like giving totally different results. And other point was the inclusion of the ions also. So that is also one of the concern during Autodoc if you are using, right? So, so you have to be selective based on different parameters.
Okay. Thank you, sir.
Okay.
Any other questions from any other delegates?
We have a question from Dr. Satish. If Dr. Satish can start the video.
Yes, ma'am. Yeah. Can I speak?
Sir. Yes, yes. Please go ahead.
Yeah. Thank you very much. Really, it was very informative and very brainstorming session for all of us. Now the world is moving on bioinformatics, and we are looking up the solution from bioinformatics. So my questions to second speaker, sir, while molecular docking, so is there any need to reconfirm again with another software? If I'm using Gromax or Schrodinger, yeah, maybe Vina. So is it necessary to check on all the software to confirm my coordination, confirmation of my ligands and my proteins, molecular docking? So could you answer on that?
Yeah. Okay, there is no clear-cut answer to this, but I think you need to verify your results. So yeah, you need to get some known data points and see if it works. If it works, that means you can use it. If it doesn't work, it doesn't use it. It's short answer. So
because a lot of attributes are there while docking and like getting ligands and studies particular study. Lot of attributes, lot of factors need to be considered. So is it necessary to again reconfirm that process, that kind of homology modeling with another software? That my question is that.
So Dr. Satish, I'll like, if you are using Autodoc, for example, for docking, so what I will suggest you, first of all, you verify your co-crystallized structure if it is docking at the same and RMSD is on the lower side. Then probably you can use the other ligands to dock in there using the Autodoc. So that is like kind of validation of the docking protocol. So you can validate it by docking the co-crystallized ligand itself. And if you are getting lower RMSD, then probably you can use it for other molecules. So this is one of the
that we can.
best thing. So, and don't depend always on the scores. So that is like which I was mentioning, garbage in and garbage out. So most of these tools, they are calculating scores. So sometimes, you know, if you'll put a very big molecule, so it automatically we can assume the scores will be on the higher side. Right. So, but we are missing the key interactions with the key residues.
That we are missing. That we are missing when you are going with the in vivo study. So we are not getting that kind of result. That we are getting the energy level we are getting over here negative, minus side, more minus side. But there we are not getting that much response, clinical response.
Yes. So those things you have to keep in your mind.
Yeah. Thank you very much, sir, for your answer. Yeah.
Ma'am, I also have a question.
Good morning, sir.
So Gajanan, please go ahead.
My question is for Dr. Vinay. Both of the sessions, first of all, I would like to tell you that both the sessions are very nice. But my question is for Dr. Vinay. Could you please tell me what's your personal view on what is the future of drug design in India? Means it is only for the publication or it is for the commercialization also?
If you will, okay, I understand that. So if you will talk about academics part, yes, we most of us, we are focusing on the publications. Yes, that is true. So because at some point we are stuck. So tomorrow you have done the docking. So for example, I'll give you an example. SARS-CoV-2 case. So we have identified some potential molecules that they may have some kind of an activity against different targets of the SARS-CoV-2. But the problem is where to get them tested, basically, for the viral studies, where to approach. So that is another limitation with the academics which we are facing. But yes, if you will look into the industrial, the research and development industrial point of view, yeah, there are, but there is a huge potential. And most of the drug discovery companies, they have their own in-house entire computational team, and they are working on these kind of things. So I personally know like Jubilant Biosys, I worked there. So we were the first, I think, when I was there, we started publishing a paper from there. And now I generally check their website, and lots of papers are coming from there and also. And they are doing those projects for other, because it's a CRO, so they are doing the projects for other companies or other bigger pharmaceutical organizations. So there is a scope. So many of my friends, they are working in other companies also, right? So Glenmark is there. So they have their in-house complete computational tool, computational team who is working on this screening and everything. And the one what we have shared you is the kind of the basic things which they are utilizing, but they also have moved ahead in more directions. Okay, so nowadays if you will see, like one molecule, if you are testing and it can have many therapeutic targets, it can, so that there also it can be, this kind of approaches can be utilized, you know. So those things are there.
Okay. Thank you, sir. Thank you for your guidance.
So we don't think like there is no
actually
scope.
I've
Scope
been
is there. Yes,
last 6
it's
years
a huge scope.
Yes, actually.
And how we utilize the things, basically. So that is the point.
Okay.
Yeah.
Thank you, sir. Thank you.
Hello. Hello.
Yes.
Yes. My question to both of the speaker, if they can suggest any open source tool for replacing Ligand Scout. Is there any tool, open source tool, which is available for Ligand Scout? It's a paid.
Exactly. Can you mention like Ligand Scout is used for what?
Yeah.
Basically?
Yeah. It is
I'm
for.
not aware about it. Yeah.
It's a
Pharmacophore modeling.
Yeah. It is basically for 3D pharmacophore modeling,
yes,
where structural molecules, even small molecules, can also be.
Sure.
So they generate heats and leads for us.
Mm-hmm. So generally, like actually for pharmacophore modeling, most of the softwares, they are basically commercialized. And if we are using also some free tool. There was one, I don't remember the name, so you just can mail me, and probably one of my student, he has used that one of the free tool. I'll send you the link and the details of that. So, but I don't know how good it was in producing the results and all the things. So
Okay.
generally, like phase is very good. Catalyst, they are good tools for the pharmacophore modeling. So and
Yeah, but
very
in
widely
Pharma
used,
the
gist,
but
I come across one.
they are like paid one. Yes.
yeah, being in an organization,
Yeah, no, that
we
is the concern most of the academic organization they are
so
facing,
sir,
and so it's a challenging thing. Yes,
yeah, it's
definitely.
a challenge, and that is why we are trying to move on to open source tools. So we have
Yes,
started
yes,
using
yes.
Gromax, and so we are focusing on basically open source tools.
Yeah, yeah.
So pharma gist I
Which,
come across, which is used for the pharmacophore modeling, and it is a free open source. Pharma gist.
yeah, that is also.
Okay. So
Tomorrow
actually,
we
I
are
was
Farmer is there.
mentioning like
Zinc
one of my
farmer is
student
there. Zinc
recently he
farmer.
worked with one of the tools. So maybe if we can get some information from there, so you can drop me a mail. I'll confirm with him and I'll send you the information.
thank you,
Sure,
Dr.
sir.
Vinay.
Thank you. We had a wonderful session, both of these speakers.
Yeah, thank you, Dr. Das
Thank you very
also.
much.
And all delegates, be ready for tomorrow's very good another session on the drug design, where the Dr. Vijay Masand will be delivering on this use of open source material as free softwares, where you can use it. So that will be a very good tool. And thanks to all the delegates for wonderful session, their active participation. Thanks Dr. Satish Polsettiwar, who is a professor here in MIT University and regularly helping us in the various techniques and tools. He is very nicely known in Maharashtra for his demonstrations to all the faculty members throughout Maharashtra for the IT tools and use of various teaching methodologies, virtual labs. Thank you, Dr. Satish.
Thank you, sir.
I have also heard his webinars, and he's so enthusiastic. So that is something creditable while listening to him. Looking at him, the other person gets motivated.
Thank you. Thank you.
This is for Dr. Satish Polshettiwar. And now let us come to our speaker. We have to honor our speaker for the nice presentation and his contribution and the insights he has given, both the speakers, Dr. Vinay, as well as his colleague. I was, told, like I had a misconception that they are Guru Shishya, but I came to know later, and I apologize for the, consideration that they are Guru Shishya instead of colleagues. So it was a great contribution, and the insights given by Dr. Vinay as well as Dr. Viswal Das. And now we have to honor Dr. Viswal Das with a certificate. I request Professor Sharad Bakode to do so. I'm presenting the certificate. Is Dr. Viswal Das there?
Yeah.
He's there.
Yeah,
Ma'am.
because
Yeah,
I'm
yeah.
unable to see his,
Yeah. I'm here.
Yeah, he's there.
Yeah.
Okay, thank you.
So we will be sending the certificate by mail, and I'm grateful to you for sparing your time at the odd time of the day and delivering the speech to all of us. Thank you so much. And here we conclude with the morning session for today. We will be joining for the afternoon session at 1:50. We have another two speakers, which are again renowned speakers. Dr. Sanjay Pai is after lunch, which would be followed by, let me just check, because hello? I just want to confirm with Vinay sir.
Yes, ma'am.
My coordinator said that I have not displayed the Vinay sir certificate, but I feel
No,
I
no,
have.
yeah, you have displayed. Yes, yes.
If, do you want me to display it
No,
once
ma'am,
again, sir?
thank you. No, thank you so much. Thank you so
Right,
much.
because I thought that I have already displayed. So
Yeah,
Dr.
it's
Viswal
worth it.
Das and Dr. Vinay have really had good insights, and which we can see from the interest which they have kindled in the mind of the participants. They all want to know different softwares which are free for them to use, and the difficulties they can resolve while proceeding ahead in their research. One question I feel, Dr. Vinay as well as Dr. Viswal Das, one question was very pertinent, that most of this work in drug designing is remaining in the arena which is not useful to the population, because the actual drugs which are coming out are very less. It is more for publication in high impact journals. So how it can be made more useful to the people, that is the question.
So, the basic point I would say, ma'am, in this case, one of the thing is the drug discovery process is very cost involving process, and you can see the situations. The pharmaceutical R&Ds, they spend such a large amount, and they ultimately come out with the failures. So, and they have to retract the molecules in the end because of the lots of side effects and those kind of things that emerge in the end. So that's why like they have started they have stopped kind of going in more and more drug discovery projects, and the things they now uh, the changes which are taking place is like therapeutic repositioning kind of approach, where the existing molecules which already have been established on the safer side, and their profiles and everything is, they know like it's safe, so they are exploring those things nowadays more. So the cost factor is one of the reasons, and the kind of earning they will be making. So that's why like it's one of the reason that they are not exploring the drug discovery projects nowadays.
I agree with you, because
Yes,
in
ma'am.
past so many years, India has come up only with 2 or 3 molecules.
Yeah.
And all this drug discovery which is happening is at, theoretical level. And second point, what I find is, we are going for this drug discovery process, but most of the, in academics especially, one student comes, he does some research, it goes in his thesis and there it ends. Another comes, takes up another molecule, or and everything ends up in the books itself.
So again, that's why like it's like the kind of a funding which are available to the academic organizations also is limited, and this thing is also one of the factor, like so we do some basic research, docking and those kind of things, do some kind of animal studies, and that's it. We end our project there itself and publish the results, that's it. So we don't go beyond that. So going beyond, we don't have the resources basically. So there we are lacking somewhere. So the possible things are like if we can collaborate with some of the bigger organizations or maybe some of the industries, so then possibly these things can go further ahead. But the research which we do at the academic level, so most of us like we are focusing on kind of the phyto things are revolving more probably nowadays. The phytopharmaceuticals, or that concept is more evolving. So if you look into the present case also, the like SARS-CoV-2, so since last 6 months, most of the companies, they are focusing on the development of the vaccines rather than the molecules. We are not finding anything so far, we have not seen. So that is also like, this shows that small molecule discoveries, really, it's a cost involving process, more specifically.
Okay, thank you, Dr. Vinay. It was very nice to listen to you. So here, can we include this phytochemistry aspect also with drug designing part?
Sure, sir, sure. So that is also being done. So most of the things are being explored. So even SARS-CoV-2 also, like if you will see, many people, they have done lots of, they have done the virtual screening of many of the phytopharmaceuticals or the phytoconstituents basically, and based on that, probably they are exploring that this kind of mechanism could be one of the factor in treating. So this, like Ramdev, also I remember, like he did also kind of I saw his paper, he has done this against this ACE2 receptors of this Vidania somnifera. He did that study. So they did some in silico screening and they published also, and later on they made that CADD, what they are proposing, coronil. In that they have included this as one of the constituent, Vidania somnifera, ashwagandha basically. So that is also there. So that is being done, yes, when many of people are doing. We have also done a kind of a study where we have screened many of the phytochemicals of, we have a kind of in-house database of molecules which we have developed, and we have screened them against all the different targets. So ACE2 also, this RDRP, RNA-dependent RNA polymerase, the protease also, the PL-Pro, and that also we have done. So we are analyzing the results. So then maybe again the thing will end up probably at the publications only, as ma'am said. So
actually, what
again, the question is because we are not able to proceed further. There are limitations.
Yes.
Better.
Yeah.
Wonderful session it was, with the two great speakers throwing light on the drug development using the softwares. I congratulate both of them. And here we end with our morning session of the third day. Let us join at 1:50 a.m. for the further sessions. Have a nice lunch. Normally, we provide a very sumptuous lunch in our institute when we carry out the physical programs, like inviting you all over here. But this time we are missing the opportunity of offering you the sumptuous lunch. But all of you have good lunch and be back by 1:50 p.m. Thank you.
Thank you very much.
So
Okay,
thank you so much, ma'am. Thank you so much for giving the opportunity to share the experiences. So I also want to thank Vees for accepting our invitation, and so.
And I loved his Madeira.
So Madeira is it's a kind of a small island which is located around maybe 500, I'm not sure, to 300 or 400 kilometers from the main island. That is the capital is Lisbon, so it is there. And one of the famous personalities from there
Ronaldo.
is Ronaldo, the footballer. He belongs to that place, and it is one of the very beautiful island, very green, and with very small population, also not big. So very beautiful island, yes.
Right. Okay then. Bye all of you.
Thank you. Thank you, Vees.
Thank you
Vinay.
so much.
Thank you.
Thank you very
Bye-bye.
much.
Bye.
Very nice to see you.